Welcome to all our participants. Today's topic is fetal origins of adult disorders. These are the objectives. One, to develop an awareness of the contribution of the early life environment to adult diseases. Two, define the term epigenetics and introduce the molecular machinery involved. Three, explain how epigenetic factors affect health. And four, discuss how misregulation of epigenetic mechanisms lead to disease states. Today's speaker is Dr. Catherine Lynn Silaw. Dr. Silaw graduated from the UP College of Medicine in 1991. She completed her residency in pediatrics at the University of the Philippines, Philippine General Hospital. She did her research fellowship in molecular genetics at the International Center for Medical Research, Kobe University Graduate School of Medicine, and soon pursued her PhD in molecular genetics in the same institution. At present, Dr. Silaw is a consultant at the Department of Pediatrics at the UPPGH, a research professor of the National Institute of Health Philippines, and head of the Molecular Genetics Unit, Institute of Human Genetics, National Institute of Health. She is a multi-awarded researcher, investigator, and national scientist in the field of molecular genetics recognized by Kobe University, USD, UP, and the NIH. May we now call on Dr. Silaw?
You could say that parenting begins at conception when a unique mixture of DNA coming from our mother and our father is born. But then this is not all you are, because how our parents reared us and what they fed us since infancy all contribute to our individualities. Now, also our mothers, what our mothers ate during her pregnancy all create critical changes that we carry into adulthood, and this may spell either health or disease, and your epigenetics now enters the picture. Now, let me first begin with a definition of genetics. Genetics is the science of genes, heredity, and the variation of organisms. And just some basic concepts. We know that our body is made up of thousands of cells, and in the nuclei of these cells are what we call chromosomes, which are the inherited material that we get from our mother and father. Now, these chromosomes, if you unwind them, they are made up of thousands and thousands of genes. Now, these genes, if you study them further, this is now made up of your double helix that was made famous by Watson and Crick. This is the double helix strand. Now, this DNA, if you dissect it further, these are made up of sequences of nucleotides or your base pairs, namely your adenine, cytosine, your guanine, and your thymine, or your ACG. Now, if let's say this is your chromatid and this is your double-stranded DNA, which, as you see, is made up of your ACGT, and this now is your normal DNA sequence, CG, CA, and the complement.
Ba't umalis si Susan? Umalis si Susan, dapat dadalhin yung food sa baba. Hilahin.
Normal gene then gets translated into a normal functioning protein. If you have a normal functioning protein, then you of course have a normal cell. A normal cell will give you a normal organ, a normal organ system, and then of course a normal function. Now, if you have your guanine mutating into thymine, so G becomes a T, then you of course have an abnormal DNA sequence. An abnormal DNA sequence will now give you an abnormal gene, which then gets translated into a mutant protein. If you have a mutant protein, then you have an abnormal cell, an abnormal organ system, an abnormal organism, and then the signs and symptoms of disease now come into play. But then this is only one half of the picture, because there is more to DNA sequences than what was said earlier. And this is where epigenetics now comes into the picture. So now, what is epigenetics? Epigenetics is the study of heritable and often reversible modifications to genes without altering the nucleotide sequence of the DNA. So there are three important concepts here, namely that it is heritable, so it can be inherited from our parents. Second, it is reversible, so it can be changed. And third, there are no alterations to the nucleotide sequence of the DNA. So heritable, reversible, and no alterations in the DNA sequence. So where does epigenetics come from? It actually was coined from the word epigenesis, epigenesis, which means on or above genetics. And this was first introduced in 1942 by a developmental biologist and evolutionist named Conrad Waddington. He believed that our phenotype, or what we are now, is a cause of is due to interactions of your genes and your environment. Now, he further said that the development and maintenance of an organism is orchestrated by a set of epigenetic mechanisms that switch parts of the genome on and off at strategic times and locations. So at different points of our are turned on and off, that makes our cells, tissues, and organs actually react to each other. So it refers, epigenetics now refers to factors that control how and when important genes can be turned on, bad genes can be turned off, and old genes should not be turned on. So next question would be, how do genes get turned off? So there are three major mechanisms that cause heritable silencing, and these are namely 1, DNA methylation, 2, histone modification, and 3, your modulation of RNA expression. So of course, the questions would be, what are these things? So let's first define DNA methylation. DNA methylation simply means that you have an addition of a methyl group at the 5' site of your cytosine. So 5' just means the left or the right side, most side of the gene. So the methyl group gets attached to the 5' site of the cytosine, but then it has to be a cytosine that is beside a guanine. So this methylation of these refers to cytosine residues when present as CG dinucleotides or CPG islands. So how do you inactivate a gene by DNA methylation? So you see here a normal gene with a promoter region, and this block are your exons or your coding regions, and these lines are your non-coding regions or your introns. So if there are no methyl groups that are attached to these white circles in the promoter region, the genes can be expressed. However, these genes can be inactivated when methyl groups get attached to the promoter regions, and these are shown here with black circles with the letter M. So when these methyl groups are attached to the promoter region of a gene, the genes get inactivated, and then eventually there is no expression that you see. So how do you apply this now to diseases, specifically cancer? Let's look at one particular gene, namely the tumor suppressor gene. So the tumor suppressor gene, as it implies, it suppresses tumor growth. So it's actually a good gene. So you don't want to inactivate a good gene. Now, there are three mechanisms of inactivating this tumor suppressor gene. First is if you have a mutation. So mutations can either be a point mutation, a deletion, or an insertion, etc., etc. So what happens is if you have a mutation in the coding region, it shortens the gene. So it either decreases the activity of the gene, or it completely obliterates the action of that gene. So that's one mechanism. Second is if the entire gene is actually deleted. So if you have an entire gene deleted, then of course there is no expression that occurs. And third would be if you have methyl groups attaching to the promoter region here, promoter region of the gene. Hence, if you have the methyl groups actually attaching to the promoter region, then this inactivates the gene, and no expression of the gene occurs. Okay. 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 Sorry, we were just having some technical difficulties. So just to reiterate, too much methylation can undo the work of protective tumor genes, and this actually happens in many types of cancer. For example, your colorectal cancer. Second, the second mechanism to silence genes would be histone modifications. So what are histones? Histones are proteins that are the primary components of chromatin. So what is chromatin? Chromatin is a complex of DNA and proteins that make up your chromosome. And these histones actually act as a spool around which your DNA can wind. So if you have, let's say, genes that you don't need, so the histones will tightly wrap these genes together, making them unreadable. So you're actually putting them into your cabinet so that they don't mess around all the other genes. So if you need the genes to be activated, or you need these genes, so the histones will now relax these genes, making them easily accessible. So that's histone modification. And third would be modulation of your mRNA expression. So this mRNAs, this microRNAs, regulates gene expression by causing heterochromatin to form, triggering histone modifications and then DNA methylation. So the key concepts here would be that the epigenome functions as an interface between your inherited genome and the dynamism imposed by your environment. And this interaction actually promotes epigenetic modifications that will create a relatively stable profile of your gene expression. And these epigenetic mechanisms actually also happen in normal cells. So how do they happen? They actually control cell differentiation through differential gene expression. As mentioned earlier, at different stages of development, there are different genes that get activated and different genes that get turned off. So this happens in normal cells. The second role would be to control transposable elements, otherwise known as your jumping genes. So these jumping genes are also called your transposons. So these are like naughty children who keep jumping around, and you don't want them to be jumping around because they might break something. And going back to genes, if they keep jumping around, then they can create havoc in the genome and maybe insert itself into another gene and then cause problems. Third would be imprinting, and then fourth would be providing mechanisms to assess the environment and adjust the genetic response. So when do these epigenetic modifications occur? They actually occur all throughout your lifetime, and they occur within different tissues and different cell types. And this is actually clearly explained by the study of Fraga et al., wherein he studied identical twins. And of course, if you have identical twins, you have the same genetic makeup. Now, when he studied these identical twins at 3 years of age, he noted that they had lower levels of DNA methylation and also identical gene expression patterns. However, at 50 years of age, these twins now had higher levels of DNA methylation and significantly different gene expression patterns. And what now happened in between? These levels of DNA methylation changed. Why? Because of your cumulative history of external factors, namely nutrition, drugs, stress, etc. And this figure actually shows it all. So you have the different environmental triggers, namely pollution, smoking, exercise, toxins, diet, infection, stress, cold, heat, and drugs. And all of these triggers actually affect your genetic machinery, affecting now your DNA methylation, your histone modifications, and then your mRNA, your microRNA modulations, and then it affects your protein, and then of course now giving you the phenotype that you have. So epigenetics stands at the center of modern medicine because of these epigenetic changes. Unlike your DNA sequence, which is the same in every cell. So these epigenetic changes can occur as a result of dietary and other environmental exposures. So here comes now Barker's hypothesis, because how early does this epigenetic change occur? So Barker believed that this actually started intrauterine, and this intrauterine exposure can result in chromatin alterations that actually persist into adulthood and may contribute to adult disease. So what did he do? He did an epidemiologic research of disease distribution, and he based this on the medical archives of 16,000 men and women born between 1911 and 1930. So he noted that babies who had increased birth weight had decreased risk for coronary heart disease. Another proof that diet actually causes epigenetic changes would be the Dutch famine. So this happened in the years 1944 and 1945. This was around World War II. And during this Dutch famine, of course, there was severe starvation and also severe winters. So pregnant women were forced to work during this period of time. And those pregnant women who were exposed early in pregnancy had babies who had more extensive disease manifestations and more severe disease. However, pregnant women who were exposed during later pregnancy had babies who had less extensive disease manifestations and less severe disease. So what else did they note? For these babies born to these pregnant women, they had increased risk for schizophrenia, antisocial behavior, and wide range of CNS defects. And female babies born had 5 times increased risk for breast CA. And interestingly, when these babies grew up in relative prosperity, of course, the Netherlands eventually had their economies improved. These babies eventually grew up and became pregnant, and their babies were unexpectedly small. So what does this tell us? It tells us that the effects that their pregnant mothers had actually now affects the next generation. So another study shows that epigenetics is also associated with perinatal stress. And they noted that global methylation levels in cord white blood cells were noted to be higher in newborns delivered by cesarean section than those delivered by normal vaginal delivery. And this high DNA methyl levels actually persisted 3 to 5 days after birth. So where do these methyl groups come from? They actually come from the foods. So foods rich in folic acid, betaine, choline, the B vitamins, and methionine, all of these methyl groups then eventually get attached to your DNA. And this DNA, of course, that then gets passed on to the next generation. So, for example, let's now use one example of one nutrient, namely your folic acid. So if you have a mother with folic acid, folate deficiency during pregnancy, they of course have inadequate levels of S-adenosylmethionine. And if this happens, you have DNA hypomethylation. And this will now cause improperly elevated expression of certain genes and genetic instability, causing now abnormal chromosomal rearrangements. And now when they give birth, the babies will now be seen to have growth defects, birth anomalies, and neural tube defects. And this is why folic acid is now advised for pregnant women or child-rearing, for women who are on a childbearing age. So this table actually just shows you the epigenetic alterations in colorectal cancer. And these are the different genes involved in colorectal cancer, namely your APC, MGMT, CDK. You don't really need to memorize this, but then these are just examples of the different genes and the different functions and the different epigenetic changes that you see in these genes when you have a patient with colorectal cancer. So this tumor suppressor gene, for example, the CDKN2A, is silenced by hypermethylation, and this will now increase the risk of your colorectal cancers. Okay. And this occurs actually, this epigenetic mechanisms or changes actually occurs in different diseases. And this table will just show you the different examples of diseases that will have the different epigenetic changes in the different genes involved in this particular disease. Okay, you don't need to actually memorize it. So next question is, can diet and lifestyle influence future generations? Of course, the answer is a resounding yes. And this also is seen, these epigenetic changes is also seen in animals. And one good example would be the worker and the queen bee larvae. So they are all genetically identical, but then what makes them different is that the queen bee, those bees that actually eat the royal jelly, become fertile queen bees, and they are now much larger and longer lived. And the rest will just turn into sterile workers. So the lesson here is to eat like a queen. For mice, you also have agoutic mice showing effects of different diets. So agoutic mice, they just refer to the yellow coat color that you see here in this picture. So a mother, there was a mother mouse that was not fed with a supplement, with a diet rich with supplements. So the children, the baby mice, actually turned out to be those here with the yellow, the agoutic mice that were bigger with yellow coat color. And these agoutic mice actually had higher risk for CA, diabetes mellitus. They were obese and they actually had reduced lifespan. Now, mothers fed with supplement-rich diet had babies who had this smaller brown-colored mouse, had lower risk for CA, diabetes mellitus, obesity, and they had prolonged life. Therefore, an individual's adult health is heavily influenced by early prenatal factors, and our health is not only determined by what we eat, but also by what our parents ate. So, next question is, how can we alter the expression of our own DNA? The answer here is to have an epigenetic diet. This is now the new lifestyle concept proposed because these epigenetic changes are reversible. Remember what I mentioned, the three important concepts I emphasized earlier. It's reversible, it is inherited, and there are no DNA changes. So these epigenetic treatments, however, must be selective to abnormal cells only with minimal damage to normal cells. So this table now will show you the different nutrients or diets at the different stages of development. And these are the third column will now show you the different epigenetic mechanisms involved. And as you see, these are the same nutrients that I mentioned earlier: folate, choline, butyrate, etc., etc. Now, one study is actually emphasizing on butyrate, which actually has inhibitory effects on histone deacetylates, and they have anti-carcinogenic properties in rat cancer models because it lowers the pH in the colon, it decreases normal colonocyte proliferation, and downregulates mRNAs involved in cell cycle apoptosis and cancer-related pathways. And this table will now just show you the different genetic and environmental factors, different genes, and this will then, of course, give you increased risk for CA. If you take green tea, you have demethylation of these certain genes, and of course, it decreases your the risk for CA. Obesity increases the risk of your colorectal cancer, of course, and then physical activity decreases risk. So why is epigenetics important? It's important because it provides mechanisms to understand how a variety of nutrients can affect our genetic background. It's also important because an individual's phenotype can be explained by both his genotype and his epigenome. It can be heritable, It can be associated with disease states, and this can be modified by nutrient factors. And in summary, the epigenome is the result of the genome interacting with the environment. Epigenetic modifications can occur throughout the lifetime of an organism, and it can modify gene expression and cellular phenotypes, and DNA epigenetics constitutes the main and previously missing link between genetics, disease, and the environment. And this epigenetic modifications actually help us better understand disease pathogenesis, and they now also provide new insights for improved treatment and disease prevention. And this is ready. Thank you.
Thank you so much for that insightful and instructive lecture, Dr. Silaw. We would like to thank our media partners, the Philippine Daily Inquirer and the Filipino Doctor, for sponsoring this event. Before we proceed to our Q&A portion, let's have one of our panelists give his reactions. He is Dr. Ed Posicion, another member of Class 91. He graduated from the UP College of Medicine and was a pediatric resident at the University of the Philippines, Philippine General Hospital, Children's Hospital of Michigan, and the Metro Health Medical Center in Cleveland, Ohio. He also took his fellowship in neonatal and perinatal medicine at the School of Medicine, Detroit Medical Center, and at Wayne State University and the Children's Hospital of Michigan in Detroit, and was a visiting fellow at the Royal Alexandra Hospital for Children in Sydney, Australia. Currently, he is a consultant neonatologist at the Iloilo Doctors Hospital, the chief of the NICU at the West Visayas Medical Center, and the unit head of the Newborn Screening Center for the Visayas region. Without further ado, may we call on Dr. Posicion.
Thank you, and good morning. Dr. Silaw made a very informative and very simple lecture on the effects of in genetics, the environment, and the interaction between the two. Now, this, her so-called explanation of the missing link between the influence of genetics or nature and the influence of the environment or nurture are now linked together through these mechanisms. It is important in pediatrics because there's now a renewed emphasis on the first 1,000 days in the child's development. And so much of public health programs, especially pertaining to pediatrics and preventive care, is now looking at the care of children, especially in the first 1,000 days. The importance of the first 1,000 days is that this is the most crucial part in a child's life, when most of the vital organs are developing, most of the sensory systems are maturing and developing as well. And this is also a most vulnerable time, because if there are significant insults to the system within this time, then this can lead to lifelong disabilities. For example, in mothers who did not take good care of themselves or had medical problems during pregnancy, diabetes, hypertension, endocrine issues, general health issues, their children may sometimes be small when they are born. This is called intrauterine growth restriction. Now, babies, for example, who have intrauterine growth restrictions, when they come out, they are small. They are small for gestational age, and we have known for a very long time that babies who are small for gestational age develop problems not only during the pediatric age group, but when even into adulthood. They are often small. They don't do very well in school. They are sickly, and when they grow up to be adults, they are not as productive as regular kids, regular babies have. And for a long time, we had been searching for reasons why IUGR, or intrauterine growth restriction, was a big problem and why the outcome is so bad. And now Dr. Silaw has provided a genetic basis or epigenetic basis for these kinds of disorders. Now, she mentioned about nutrition and the importance of nutrition not only during the time of pregnancy, but perhaps nutrition is just as important after the baby is born. We have always looked at nutritional rehabilitation, for example, of the preterm baby, and nutritional rehabilitation of the small for gestational age babies as a way for them to catch up with the rest of the population and perhaps have the same outcome as the rest of the population. But we've always discovered that there are significant differences in terms of development between the small for gestational age baby and the appropriate for gestational age baby. And this is probably the reason why. The other interesting thing about small for gestational age babies is that long-term longitudinal studies have reported that they are at risk for that more of them develop coronary artery disease, hypertension, and hypercholesterolemia later on in life. And therein comes Barker's hypothesis, saying that because the babies were nutritionally deprived inside the mother, their physiology has been altered to the point that when you nutritionally rehabilitate them, they actually do badly. So although the intention of pediatricians and neonatologists for a long time was to give lots and lots of nutritional help to make the babies anthropometrically catch up with the rest of the population, what Barker's hypothesis is now saying that these kids will develop hypertension, diabetes, and coronary artery disease largely because of the perinatal or antenatal insult to their systems. So are they doomed to have it forever? Will they always have the same disorder, or are there things that we can do to actually prevent this from happening? Therein now lies an important question to Dr. Silau's lecture. If epigenetics is a significant influence in the development of coronary artery disease and hypertension based on Barker's hypothesis, then there must be other interventions. There are other interventions that we can do to prevent this from happening. And she mentioned it towards the end of her lecture. Folate supplementation is important, not drinking alcohol, having a healthy diet, and not being obese. May be good interventions to modify these epigenetic changes in the gene and perhaps prevent the manifestation of these disorders. So we now stand at a threshold in pediatrics and in neonatology in the sense that pediatricians are not only involved in well care, in early stimulation, in nutritional advice, in vaccination, but now they should be involving themselves more and educating themselves more towards a healthier lifestyle, even for their pediatric patients. Indeed, we cannot only say that the healthy lifestyle is for the adults, for the much older generation, but a healthy lifestyle must start actually even during pregnancy and must persist right after the baby is born and throughout the baby's life, throughout the neonatal period and throughout infancy, childhood, and even into adulthood. So I think those are just my main reactions to her lecture. I hope that our fellow pediatricians realize the importance of programming, and that they cannot only focus their attention on anthropometrics, well child care we call it, but they must also focus nowadays on even healthy lifestyles for their pediatric patients. And whenever they see their pregnant mothers coming to the clinic, they must reinforce the obstetrician's advice for a healthier lifestyle. Perhaps in the future, epigenetics can also be used to treat existing disorders, since it is reversible. If we can perhaps determine the actual epigenetic insult in the gene, then we can use epigenetics also to probably modify the disease. Again, we would like to emphasize that the first 1,000 days in a child's life are the most crucial, because this is the time when the brain growth is maximized, sensory stimulation is most important. This is the time when the child actually develops most of his mental abilities. And if there's any deficiency during the first 1,000 days, then that can lead to a lifelong deficiency already. For example, if a child is below the third centile for height, by the age of 2 years, if the child is not caught up, then he will always be short. And much the same with the other organs. I think that's all I have to say. Thank you very much for giving us this opportunity to share our knowledge and our expertise. And we hope that you learned a lot from this webinar. Thank you.
Thank you very much, Dr. Ed Posession. That was a very engaging discussion. We now have time for some questions for our speakers and our speaker and the actor. We have one question from Dr. Kathleen Haynes. Her question is, could you please comment on the claims of most of the milk formula companies that they are now at par with breast milk? Dr. Sila?
I don't really know the components of milk formulas. However, what I know is that human milk has lactoferrin, prostaglandin, the PUFAs, and undigestible oligosaccharides. And these lactoferrins actually, some of these components actually are very important in preventing necrotizing enterocolitis, correcting or preventing immune disorders, obesity, gut problems. And why? Because there are certain genes that are expressed with this particular human milk components. But then for the milk formulas, I don't really know whether they actually have these same components that breast milk has.
Dr. Posession, would you care to react?
Yes. In fairness, the science of infant nutrition using infant formula has advanced itself by leaps and bounds over the last decade. Increasingly higher caliber research is being done to almost copy. I'm saying almost copy because they will never be able to copy or reproduce human milk. But they are getting closer and closer to copying the essential nutrients and components of breast milk. What they can never provide, as of now anyway, are the more bioactive components in breast milk. You know, the immune modulators, for example, the antibodies that mothers transfer to their babies through breast milk, that cannot be transferred, that cannot be placed, that cannot be put as an ingredient in formula milk. So although the science of formula production has improved and trying to copy all the ratios and all the percentages in terms of protein, sub-protein components, even antioxidants and DHA, AA, and all those things. These are, as you say, ingredients in a cake. Ingredients, which is like making a cake. You put the ingredients in and you have a cake. But human milk is really a biologic fluid. It is live. It is a living substance with living components in it. And that cannot be copied. And so they may get close, the formula companies may get close to the ingredients of breast milk, but to replace breast milk in itself is an impossible task for a formula, as of now anyway. So nothing will beat breast milk in nutritional value, in long-term effects. You know, we have a lot of studies already indicating that breast milk is beneficial, not only for mental development. Kids who are breastfed longer have higher IQs than those who are not breastfed, for example. There's a lower incidence of asthma, diabetes, hypertension, even cancer in babies who were breastfed. And the longer the mother breastfeeds, the more significant the effect, the beneficial effect on the baby. So at this point, there is no substitute for breast milk. as a not only as a nutritional source for babies, but also as a form of protection for future adult disorders.
Thank you very much. Our next question is there a role for cord blood banking in epigenetics?
I don't really know whether there is a role of cord blood banking in epigenetics. Actually, probably I'm just speculating that if you bank the blood, there may be methyl groups that can be attached. I have no idea, so I really have no idea how to answer that question.
Dr. Position?
Right now, cord blood is very popular. Cord blood banking is popular for various reasons. One, the onset or the use of bone marrow transplantation, for example, in leukemias and in other disorders, has looked at cord blood as a source for germ cells, for pluripotent germ cells. And so it has gained popularity because somehow if you've had your baby's cord blood banked or stored, in the event that a child should develop a disorder wherein bone marrow transplantation or just stem cell transplantation is curative, then you are protecting the child from that disorder should it happen. So, and there are other applications for cord blood banking, but in pediatrics and in neonatology right now, cord blood banking has become popular largely because of the pluripotent stem cells that are found in cord blood. But of course, you know, you're storing something for the future. You never know in the next decade when somebody discovers, for example, a way to, they've already started it, a way to regrow your liver, no? And they'll just get your cord blood from the bank and they will regrow your liver as an adult and put it back to you. They will regrow your skin if you get burned. If you need a skin transplant, they can just take, again, pluripotent stem cells from the cord blood that the baby donated and is now an adult, and they can use that to regrow new skin, new muscle, new bone, whatever other cell that your child needs can be regrown into something that will benefit. Of course, there are ethical considerations to that. You know, can you clone a child from cord blood, for example? I don't know. Maybe Dr. Arcila can answer that. But right now, the popularity of cord blood banking is its ability to somehow protect a child from future, should future disorders come and this technology is available, then it is a source. Rather than rely on organ donors, for example, you can just transfuse, you can just use the stem cell as a source for curative technology or curative therapies for these children. Other than that, right now I'm not aware of cord blood components being used somewhere else, other than the storage of pluripotent stem cells or other components for future disorders.
May I just mention that we had 72 attendees with 12 people on live streaming. We have yet another question regarding autism having possible fetal origins. Would you care to comment on that, Dr. Position?
We don't exactly know. We know, for example, there was a large autism convention in Australia about 2 years ago, and they looked at the genetics of autism, and they seem to have found the genes for it. That is one of the important breakthroughs. But whether the gene truly manifests or there are epigenetic factors that can alter the manifestation of autism as a genetic expression, I believe it up to the geneticists to figure out. But they have described the genes which seem to be the one that's associated with autism. I think the next step now is to document, for example, to see, for example, if we can modify the expression of the gene through gene therapy, for example, or from other means, for using epigenetic factors or other technologies to prevent that particular gene from manifesting. Or if the gene is present, as we know, is there something that allows it to manifest? Is there a biological hit, they call it? Is there an influence that makes the gene manifest itself? Or the reverse, are there factors like nutrition, vitamins, or whatever that can prevent the gene from manifesting? But as far as I know, they have, but based on that Australian conference anyway, they have identified the autism gene, or the group of genes actually that seem to cause autism.
I agree with Dr. Precision that nutrition may actually be a cause, a factor that people need to look into, because I attended the genetic conferences abroad, and actually they have been doing GWAS studies for several thousands of patients with autism. However, they have not really agreed as to what particular gene is the causative gene for autism. So they still are probing into this. However, they're now looking into the metabolic factors or nutrition as possible causes for autism, and maybe this is also something that people should be looking into.
Okay. Dr. Silaw, what kind of interventions would you usually recommend in such patients with genetic diseases?
Interventions, well, generally, of course, we need to find, they need to be seen by a geneticist to find out whether this is really a genetic disorder or not, and to find out the actual diagnosis. And then a geneticist now will then decide what particular tests to do, whether they can be done in the Philippines or whether they need to be sent out abroad. We do have a molecular genetics lab, and we do have certain tests that are available now. However, these of course are based on kits that are using results from populations abroad. We are now finding out what are the common mutations in our populations, and then we do hope to develop our own kits for our populations. Now, for epigenetics, I guess, again, we go back to how avoiding stress, because stress can actually add methyl groups to your DNA. Eating well, as Dr. Precision mentioned, because this also can increase the effects or the expression of the good genes, and also reduce the expression of the bad genes. I guess that's really.
Thank you very much. Any last words, Dr. Precision?
If I may add, the future of newborn screening, for example, is towards genetic screening. As you know, newborn screening has existed in the Philippines since 1996. It's been 20 years, and every year we come up with newer technologies and newer knowledge on how to screen kids, on how to screen babies for an increasing number of disorders. And these are all genetic disorders. Perhaps the future of newborn screening in our society would be our increasing ability to detect increasing numbers of genetic disorders. Not only metabolics, not only hematologic disorders, but perhaps the detection of genes that lead to mental retardation, to genetic mental retardation, and perhaps the detection of genes that lead to other disorders, diabetes, hypertension, cancers. Certainly, there have been many studies already looking at newborn screening samples and looking at them again and looking for markers for leukemia, for example. This was done in Michigan a long, long time ago. And they reported, for example, that using newborn screening samples as a way to detect the prevalence of certain biomarkers for leukemia can be used as a screening tool, as a way of preventing, as a way of increasing knowledge, and as a way of early monitoring kids who are prone to develop these particular types of disorders. So one of the future endeavors in newborn screening in the far future, probably at this point, is the detection of potential risky disorders like leukemias, tumors, and cancers that have very strong markers that can be picked up on a filter card, on a blood spot in a filter card. So we have a very strong newborn screening program, and we hope that in the future this will be one of the endeavors that the newborn screening program will go into, and is actually being considered in other newborn screening programs in other countries. So there is hope. There is some future direction in our abilities to detect diseases early, even from a genetic level, and with more knowledge on epigenetics and the factors that come into play, perhaps we can be more scientific in our approach to these disorders. Now using the power of genetics to cure, not only the power of genetics to treat, to evaluate, and to diagnose, but using genetics to cure, maybe the future also of medicine.
Thank you. I believe our listeners will agree with me that this has been a very engaging discussion. To summarize, epigenetics is a fairly new but exciting field which may hold the key to management and prevention of many adult diseases. Currently, non-communicable and lifestyle diseases are already on the top of our list of causes of mortality and morbidity, and definitely a lot more studies should be done to further promote healthy lifestyle among Filipinos to ensure that we get the best of health. After this session, an email containing a survey link will be sent to you. After answering the survey, your certificates will be sent to you. Please answer the survey so we can assess our webinar and address more of your preferences and give you materials for this session. Thank you again for joining us. We hope to see you again during our upcoming webinar on August 31, the last Wednesday of this month, from 12 o'clock to 1 o'clock Manila time, with our speaker, Dr. Lorraine Bangayan Abiad, on the management of resistant hypertension. Please invite your colleagues to join us in this continuing monthly CME webinar series. All webinar schedules and resources will be posted at www.upm.edu.ph/UPMedWebinars. In behalf of UPCM Class 1991 and the UP Medical Alumni Society, we also thank our collaborator units, UP Manila Information Management Service, National Telehealth Center, UP College of Medicine Postgraduate Institute of Medicine and Medical Informatics Unit, DOST ASTI, and Ms. Charis Orhalo, our host, and happy birthday. We would like to also thank the Philippine Daily Inquirer and the Filipino Doctor for their support as our media partners. This is Dr. Esther Sagal together with Drs. Lynn Silaw and Ed Posicion and the UP Med webinar team closing this session. We hope you have learned a lot from today's webinar. Join us again on August 31. Thank you, and have a great week ahead.